So Bento is a project that stemmed out of working with mini-scope imaging data. And I think in general in neuroscience, there are lots of these data sets that people are collecting where you have a lot of different modalities of data and they're assembled at different frame rates. Maybe you start acquisition at different time. And we wanted a way to kind of visualize everything side by side. One of the big issues with analyzing neural activity from freely behaving animals is you can only really analyze what you've annotated for. If you have manual scoring of some behavior, you can look at neural responses to that behavior. But it could be that neurons are responding to something else that your animal is doing that you didn't think to annotate for. And unless you see neural activity and behavior side by side, it's really tough to find those kinds of correlations. So when you're tying together different data sets, there's kind of two approaches that you can take. One is to create a specialized file structure like Neurodata Without Borders, where all of your different data formats are kind of lumped together. The other is to store paths to the different files associated with an experiment and load things from those files. And this is what we do in Bento. Bento is a MATLAB interface. It has two parts to it. It has an experiment manager and it has a data visualization GUI. And this is just an image you can find on the wiki for the Bento kit of the Bento GitHub. Bento's experiment manager is a way of entering in information about the different data types associated with a particular experiment that you're working on. And then Bento's user interface itself is a way of browsing through synchronized data of different modalities. So it can handle videos of animal behavior, manual annotations, which can also be used to generate traces of neural activity. It can also plot spectrograms of mouse ultrasonic vocalizations. When you create an experiment in Bento's experiment manager, which we'll do in a second, you track things in terms of which animal you're imaging in, the session of that imaging, the trial within that session, and then some file paths to data associated with a particular trial. Bento loads all of this data into a standardized structure format within MATLAB, where each trial, so this is mouse number 204, session number 2, trial number 3, has a set of fields storing all of the data types from your experiment in a way that you can go in and access them, both for your own analysis and in a way that Bento is able to display them within the Bento browser. So what I'm going to show you today is how to set up one of these experiments. Well, first how to download Bento, how to set up an experiment using the experiment manager, and also how to work with the experiment file outside of Bento. And then we'll go through visualizing things in Bento and some of the analyses and processing that Bento supports. So stepping out of PowerPoint now. This is the GitHub page for Bento. I'll post it in the chat as well. If you don't have a GitHub account, I warmly recommend it as an easy way of keeping track of this repository, being able to pull updates, and also being able to post issues. So within the GitHub page, you can do a couple things. You can click this code button and download the Bento repository as a zip file. If you do that and then unzip it and then just drag that directory into your MATLAB working directory, you will be able to add it to your MATLAB path and then run Bento from the MATLAB terminal. There's also this issues tab here where you can report issues with the interface. You won't be able to use this if you don't have a GitHub account, but if you sign in and click new issue, you can post, as you can see, we have a couple of these error reports of things that are currently broken. And this code is kind of thrown together over time in MATLAB. So we're really interested in where things are breaking for people. We're also support for file types that aren't currently usable within Bento. So when you download Bento, you'll get a repository of files. So this guy. Another strategy that I recommend is to install GitHub Desktop. That's a repository manager for GitHub repos. So if you have Bento and you clone it into GitHub Desktop, you can just go to your Bento mat repository. It will tell you any files that you've updated, or it will give you access to the repository in your browser. So you can open up the GitHub repo for Bento, you can find where all the files are, you can see what's going on there. And if there are updates to Bento, to pull those updates, all you do is click this Fetch Updates button in the corner. And if anything has been pushed, this will change to a Pull Updates, and you click that again, and it will sync your local copy of the code with the code that's on the Bento GitHub repository. So as we push updates to the code, you'll be able to pull those to your local computer without having to go to the web page and re-download the zip file. So once you have your repository cloned onto your local computer, it'll be in some path on your computer. So in my case, it's in my GitHub folder. It's not that guy within Bento mat. You have a set of files. To gain access to these files from MATLAB, you need to add this Bento folder to MATLAB's search path. So you need to tell MATLAB to be able to find it. So to do that, we're going to type add path and then provide the path to this folder and enter. So now MATLAB, when it's looking for something, will know to search within this folder for something called Bento. Actually, what I recommend is doing add path and then gen path. So that will add not just this folder, but all of the folders inside of this folder so that any function within Bento is immediately accessible. So once this GitHub folder is in your MATLAB search path, then to launch Bento, all you do is type Bento in the command line. That will bring up this super professional looking GUI of the Bento interface and give you a set of options. We'll do new experiment first. This is what brings up the experiment manager. And the way that Bento organizes experiments is it assumes that all of the files associated with a particular trial or with a particular animal are within one parent directory. So this is a directory somewhere on your computer that holds all of your experimental data. So in this case, I have a folder on my desktop called Bento demo, and I'm just going to look at data from this one mouse 457. So I'll open that and make that my parent folder. And then all of the files that I load within this parent, that I associate with this experiment, are within this parent folder. So to populate the experiment manager with files, I'll launch this file finder, and then I can search for a particular file name type, or I can just hit go, and it will list everything that's inside of this directory, including subdirectories within the directory. It's a lot of files. So in this case, we're going to look at this movie here. So this is an odd movie format, this .seq, that my postdoctoral lab used. You can use .seq movies, you can use AVIs or MP4s, anything that your computer has a codec for. So we're going to click on the name of the movie, then click in the behavior movie field and hit add. If we want to also look at, say, a front view video from the same experiment, you can click on that movie's name, click in the behavior movie field again and hit append. And what it's doing in here is it's appended, it has the relative path to the top view movie, and then it has a semicolon and it has a relative path to the front view movie. By default, Bento stacks these two on top of each other. If you want to display them side by side, the clever, dumb hack is just put a second semicolon between them. So now we have behavior movies. And then say we also want to look at manual annotations for this pair of movies. So I want the experiment that was done at 1800 hours, 6 minutes and 26 seconds on this date. So I'll put in that in my search string and hit go. And these are all of the additional files that are associated with that movie. So here are two movie files themselves. And then in this case, we also have down here some output from our mouse tracker Mars and some behavior annotations. These are audio outputs. Let's see, these are the finalized annotations behavior. So we'll click on one of those files, click in the field of the manager where we want them to go and hit add. And you can do the same for pose estimation data. Bento can display pose estimates from our own software Mars. It can also handle output from Deep Lab Cut and potentially Sleep. If you have imaging data, you can add that here. You can specify frame rates for those two for the imaging data and for the annotation data. And you can add additional trials from a given mouse, or you could say introduce a second mouse in your data set and renumber your trials. Bento will keep counting up from wherever you left off. So you have a second day of imaging in this mouse, you can add trials like that. You can also click on a given trial and hit duplicate, and it will make a second trial that's starting from however this first row of the database was populated. Once you're happy with your experiment setup, you can click Save Info here, and it will output the contents of this experiment manager into an Excel file. So we'll give it a name and hit Save. And if we look inside our demo directory, we have somewhere in here, my new experiment. We can open this up in Excel and we can see that Bento has output a bunch of experiment data into a spreadsheet. You don't have to use the experiment manager within Bento to populate this. You can also edit these things directly, which might be a little bit easier if you have a lot of trials that you'd like to put into a single session. The other thing to know is that by default, it populates the sheet with the parent directory of the project in this upper corner. But if you find that you're moving things around a lot or you're accessing the same project files from different computers, then so long as this Excel sheet is in this directory here, you can delete the contents of this file, of this field here. And when you load this Bento experiment sheet in the future, Bento will just look for all of these files here in the same folder as this Excel sheet. So it uses the location of the Excel sheet as a parent directory instead of the value in this field. So we've populated some fields of this experiment sheet. I'm not going to save those changes. For the sake of demoing, I have another version of the sheet with a little bit more information in here. So as you can see, we have a mouse number. This was just the ID of the animal in our database. This is the second day of imaging from this animal. We had 4 trials where the animal was encountering different stimuli. And then we have some calcium imaging data. Bento doesn't just display calcium imaging. You can put any kind of neural recording file in here. It just is named calcium imaging file. So as you see, all 4 of these fields are the same imaging file. And that's because when we did trace extraction from our experiment, we concatenated several movie clips into a single file. So what these start CA and stop CA frames or cells are doing is saying the first trial in this experiment is associated with frames 355 to 14,000. The second trial is for these set of frames, the third is from these guys, and the fourth is from these guys. If for some crazy reason your different trials have different frame rates, you can specify that here. Otherwise, you can just put a master frame rate here. And the same thing, we have annotation files. If you have multiple trials in an annotation file, you could give start and stop frames here. This offset frame gives an offset time in seconds between the start of a behavior movie and the start of your imaging data, where a positive value means that your behavior movie starts before your calcium imaging. This is tracking output from Mars. You can load audio files. This is currently unused, this TSNE here. So when you create an experiment in Bento, you can either load it from the browser by clicking this import button, or you can click the load experiment button and open it from a folder on your computer. So we're going to go in and load that Excel file that I just showed you and hit open. It will go through some time of loading things. Sometimes you'll get some warning messages. If you have tracking data, it's going to ask you how the tracking data is formatted. And in this case, we used the Anderson Labs mouse pose estimation system Mars. So we'll hit OK there. And in a second, it will load in your imaging data and your behavior movies and your tracking and annotations. So check the chat to see if we're going OK. Oh, all right. So this is the, I'll answer questions at the end, but do keep posting them in chat. This is the interface for Bento. Video, these are traces of neural activity to the side here. Pose estimates are superimposed on the video, and annotations are displayed along this scroll bar here, which is tracking the experiment time. You have a legend here of all of the behaviors that are in the annotation file. The current behavior is displayed in this corner, and behind these neural activity traces is also annotation data, so lots of places. The neural activity can be displayed in different ways. I'm plotting now as lines. You can plot as an area under a line. Looks kind of ridiculous for data sets with a lot of neurons, but if it's a fiber photometry trace, this can show level of activity. You can also display it as an image where the color is the level of activation of the neurons, which is what I tend to do if there's a lot of data here. The last one is kind of experimental and a little broken. This is just a spatial map of the neurons, which will eventually be a field of view from a Miniscope movie. And here we're seeing neural activity traces, annotations, is this guy, and the behavior video. One of the things you'll notice is that sometimes Bento breaks. If it breaks, the interface becomes somewhat unresponsive. You can't operate the scroll bars. If this happens, which I'm sure it will, type Bento in the command line, and it will somewhere pop up a prompt. What's hiding behind Zoom? It'll pop up a prompt that looks like this. It says keep data currently in Bento. If you hit yes, it'll start running again where things left off. So in this interface, you're seeing a handful of windows displaying different types of data. You can toggle other views by clicking this display menu and clicking toggle object visibility. One of the most useful is this find a note, which shows annotations of behavior in multiple channels within a smaller window of time from -20 to 20 seconds. There is an active channel, which is what's displayed on the scroll bar and is listed here in this channel menu. Each channel can have multiple behaviors annotated, and you can annotate multiple behaviors at the same time. Different channels could be used to separate things out, like the actions of one mouse versus the other, or behavior versus stimulation times, and kind of gives you more flexibility in what you display. To create your own annotations within Bento, you can add channels, give them names, and within each channel, add behaviors. When you add a behavior, it will ask for a hotkey. So once we've added this behavior, we can now annotate for it in the video. So we'll scroll to where our behavior is happening. You press the hotkey that you assigned, click and drag, press the hotkey again, and it creates this block of time that's now annotated for our new behavior dancing. You can edit the hotkeys for all of your behaviors, and you can edit the colors that are used for behaviors just because. So we'll click on dancing, we're going to make it orange. As you're annotating, thank you, Citrix, you can use Bento primarily just as a behavior annotation tool. So load up your videos, scroll through it, annotate for what the mice are doing. You hit the save button here, and it will output the annotations that you created into a file. So we'll give this a name like this, and hit save. These are called .anote files, but what it really is just a text file. So you can open this with something like Notepad and see the way that the annotations are stored within this file. So up at the top here, we have some information about which movie was annotated, the frame rate of the annotations, list of channels, list of behaviors annotated, and then in each channel, the start and stop frame and the duration of each bout of behavior. So this is what we loaded in to get this initial set of annotations here. And that's something that you can create from scratch within Bento if you don't have annotations in the first place. Right now, Bento can load annotations from a fairly small range of formats. I think we have support for Bento itself, for Caltech Behavior Annotator, and possibly EthoVision. If you annotate or record neural activity in some different format, we can pretty quickly add Bento support to load that annotation format. We just need to see a sample of how the data is formatted so that we can write a loader for it. A handful of other useful things to do within the Bento interface, and then we'll get into just opening up for questions and talking about the data that Bento supports. Within these menus here, a couple really basic things in display. Well, let's see what we want to do. You can do things like scale the brightness of the movie, some basic creature comfort things. You can zoom your movie to a particular field of view and revert it. One of the most useful things for working with Bento is just being able to save frames from behavior movies and save clips of time. So if you click Save this movie frame, we'll go to our demo directory and hit Save, and it outputs the current frame of video, like so, with the pose estimates if you have them and with whatever's happening at that time. And you can also save movie clips. So to do this, you'll pick a save destination, pick a file format, do MP4, hit save. It'll pop up this movie settings interface. If you pick MP4, make sure that you make your quality settings MPEG-4. There's some display options like the background color, whether Bento displays this scroll bar of time, the playback speed, you can give it a title, and then the segment of time that you'd like to save. So we'll start at the current time, 2 minutes and 41 seconds. We'll save till 5 minutes. We'll sample it at 30 Hertz. And one of the other nice features of saving is you can either save an entire segment of time, or you can save only the clips that are annotated for a certain behavior. So if we click approach, it will only save the clips of time that are annotated for approach. So once this is all set up, we're going to hit go. We did custom titles, so it'll ask us for a movie title. So let's call this Bouts of Approach and hit OK. And Bento is going to convert the interface. So this is now the process of writing movie clips. It's changed the background, it's turned off some of the buttons, and what you're seeing within this window here is what you're going to get in your movie output. And I'm realizing now that the black background means you can't see the legend or the title, but you get the idea. So it's scrolling through all of the annotations of approach behavior that happened within the window of time that we specified. It's adjusted the bounds of the scroll bar to only contain the segment of time that we're saving in the video. So this gives an overall summary of what's happening in that window of time, and this gives, again, the zoomed in version. It will finish doing its thing, and once it's done, revert back to its normal appearance. And if you go to your directory, we now have our movie saved. And it plays back at the full 30 Hertz frame rate. And as you can see, it's just showing the periods of time where this approach behavior is taking place. Okay, next things. One last display option is the window time. It's currently set to 20 seconds. We can change this, and it will change the time relative to the current frame that's being displayed in the neural activity trace and in these annotations here. So this dashed line is the time corresponding to this frame of video and this annotation. And here you're seeing what's happening around this time. If you are displaying, oh, in addition to displaying images, you can display different processed versions of your traces. You can apply things like z-scoring to your traces, which can make it easier to see what's going on. This is just normalizing it so each neuron has a mean of 0 and a standard deviation of 1. And in addition to plotting traces, you can, I think, plot the first handful of principal components of the data, if I haven't broken this. So we're going to exclude the last 2 trials, compute the first 5 principal components, and hit OK. And we'll convert this back to lines to see what those principal components are doing over time. We can also plot this as areas to see, and we'll z-score these. to see the deviation from baseline of the activity projected onto these different PCs. So we'll go back to image and go back to units. Handful of other options in here. Let's see. In addition to these display settings, the ugliest interface in Bento, you can pick which neurons to plot. This is super dumb. It just lets you focus in on a subset of your cells. You can also pick which annotations you're showing by toggling them on and off here to show and hide things from this display bar here. You can also do that just by clicking on these boxes here. And then we have a set of plugins. So the goal of Bento going forward is to make something that's, like I say, kind of ImageJ for these kinds of data sets. So once the data is loaded into the standard format that Bento uses for displaying, we can pass data packaged in that format to other downstream functions to process it. So for example, we have, some of these are in progress right now, but the main ones that are interesting are k-means clustering. So we'll pick a trial to cluster on, pick a number of clusters, decide whether to z-score our traces and hit go. And it groups neurons by their response types over the entirety of the trial. You can also cluster based on the activity of neurons during a particular behavior. The other plugin is the behavior triggered average. So in this interface, you pick a behavior, let's say mounting, and you can look at the activation of cells within a window of time relative to that behavior. So we're looking from 10 seconds before to 10 seconds after mounting behavior. We can discard short bouts or merge bouts together if they happen close in time. And then we can so we're triggering on the onset or the end of mounting, and we are looking at either the population average response, or you can scroll down and look at what individual neurons are doing triggered on this behavior. So it's a quick way to get a sense of what's happening in your data and what your neurons are responding to. If you want to work with this data outside of this interface, you can save a figure, which will save these two panels here. This is the average across trials of mounting, and these are the responses of unit 10 on the individual trials. You can also click Save BTA to Workspace. You can pick which things you want to work with. You could even select all of them, all of the neurons, and hit OK. You give it a name and a time axis, and then you'll have access to it within your MATLAB workspace. So while Bento is running, there's no access to the command line here. So to get access to these variables, you'll need to hit Control-C to break out of the online version of Bento. And then if you look at this variable, you'll see we have this ETA, which is a structure. It has a field unit 10, which is this neuron. And you can do things like look at the activity from the cells. So just do figure C of B. And these are the delta F over F traces for this unit triggered on the onset of behavior. You can also use the time axis that was exported. So now we have time relative to the mounting onset in seconds here. The last thing I wanted to share with Bento is that you can use it without using the interface itself, just as a way of loading in your experimental data. So in the command line, figure this out, what we'd like to do is load the data that Bento is displaying here into MATLAB itself so that we can work with it as a variable. So we'll do experiment equals load experiment. And then we're going to give it the path to this Excel sheet that we used to load the data into Bento and hit enter. It will go through the same loading process that it did to load things into Bento, and it will return everything into a MATLAB structure. So if you look at experiment 457, which was our mouse ID, it has a field for each session, which is say a day of imaging. And then each session is an array of MATLAB structures. So if we wanted to look at the first trial, we would do that. And here's the data set from our first trial. Here's the raster that's getting displayed over here of neural activity. CA time is the timestamps of those frames of the calcium data. Anno time is the timestamps of your annotations, and the annotations themselves are within the structure and note. So if you do dot session 1 or dot session 2 trial 1 dot a note, it has 2 channels. You can load those channels, and we see the annotation bouts for all of the behaviors taking place in the data set. So if you look at, say, mount, these are the start and stop frames of each mounting bout. So start here and stop here. Frames are corresponding to times in this vector anno time. So you can use the frame numbers to get back a time at which the behavior bout occurred, and then write your own code to see to analyze what the neurons are doing during that time. Let's see if there's anything else here. In terms of data formats that Bento supports. So we'll close it up here. You can put anything you want in these file paths, but if your data isn't in a format that Bento expects, it's not going to know what to do with it. Calcium imaging files, it currently takes .mat files with the output of CNMFE, which is a tool for calcium trace extraction. If you use some specialized software to extract your calcium traces, you can also just save them in a mat file by themselves as a matrix. So if I say I have some data, here's just a random signal. Say we have 100 neurons and 10,000 time steps. You can save this to a mat file. I remember how to do that. Give it a file name. And then if you put this file into your calcium imaging file path here, it will load it as imaging data. Annotation files, it's a little more picky. It needs to be a format that Bento knows how to process. Behavior movies, any standard movie will work. And tracking data, currently Mars output and DeepLabCut output will work. So I'm going to wrap up the formal part of the tutorial here. There's a couple questions in chat that I'll look at, and then if folks have other questions or requests about supported file formats, you can take those here. Okay, going down from the top. Yeah, the manual list of animals. When you create this Excel sheet here, we don't have a good way to populate this automatically. We're trying something like that for the Python format, but it's tricky because everybody sets up their experimental data a little bit differently. You can make Bento files that have a lot of different trials in them. I might have an example here. Pull it up really quickly. Sec. Oh, okay. I might not have it handy. I'll just say that you can add data from multiple mice in here. You could add a mouse 458, 459, and populate that data. When you open the Bento user interface, there are drop-down menus to choose between different mice. So you can pick which mouse you're displaying and pick which session and trial you're displaying. Multiple channels as separate files for the same experiment. If it's neural data that's not currently supported, you would need to be able to concatenate those channels into a single file. We support multiple behavior videos by appending them into the experimental loader. We also support multiple annotation files, concatenating them in. And we haven't had this case of multiple files containing neural, what you'd load in as calcium imaging traces here. So it's not currently supported, but it's something that we could add if there's enough interest. This question here, difference between an annotation file and a tracking file. Annotation files are the start and stop times of behavior bouts that have been manually scored, like I showed you for the example .note file. Tracking is the output of something like Deep Lab Cut, where it's pose estimation tracked key points on the body of a mouse. Deep Lab Cut is most common, but we support other pose estimation formats as well. For wide field imaging data and other data formats, okay, lots of questions on here. You can display the traces so long as you're able to export those delta F over F traces into some format that Bento is able to recognize, like a map file that contains a single matrix. If you want to see the movie itself, so long as it's the whole like field of view of cortex alongside the traces, you could put that into the behavior movie frame right here. And so long as it's a standard format, then you can load that in. There's a couple questions about generating annotation files and behaviors. So what I'll do is show the annotation process from scratch. Currently, we don't have any examples of Boris formatted annotations. If you send us an example Boris file or a script to load Boris annotations into MATLAB, we can add support for Boris into Bento. I suggest posting an issue on GitHub so that we can track that, but that is something that we are aware of. If you don't have Boris, we'll pick a movie, load this in very quickly just to have something to annotate. And hit start. So say I wanted to annotate this movie and I don't have anything loaded already in existing annotations. I'm going to hit add here and create a channel and hit OK. It creates this legend space for the annotation channel. We now need to add at least one behavior in here. So give the behavior a name, give it a hotkey, and it appears here. Now that we've done that, I'll turn on this find a note. You can click and drag this to track through your video. When your behavior is happening, you hit your hotkey to start annotating. You click and drag and hit your hotkey again to stop. Do this multiple times. If you hit backspace and click and drag and hit backspace again, it'll clear annotations. If you want to add a second behavior, it will create it for you, and then you can annotate for sniffing using the sniffing hotkey and following using the following hotkey. Once you're happy with your annotations, you hit save. Save it where you would like them to go. It will give it a default name, which is just the mouse number, session number, and trial number, but you can call that anything. And now if you open the .note file that was just generated with something like Notepad, it will give you the times of the behaviors that you annotated. And in the future, if you wanted to load this back into Bento, you would just add this to the annotation field of the interface. So annotation is pretty quick and painless. There's also this fast edit button. Oh, I should say you can jump between the start and end of behavior outs by hitting page up and page down. Play the movie by hitting spacebar. Up arrow doubles the playback speed, down arrow halves it. And if you toggle this fast edit and go to the start of a bout, you can relabel bouts just by hitting a hotkey or pressing delete to quickly change the label of the behavior. Other questions? Let's see. The best way to retrieve processed Bento data, behavior triggered averages I showed. For things like the z-scored traces, I suggest using this experiment loader. Right now, it doesn't apply any of the processing of things like clustering when it loads the data. It just loads it as it exists in the raw file. But if it would be helpful to output things like cluster assignments to the MATLAB workspace, we could set that up. Currently, the only thing that we output to the workspace is this behavior triggered average, but that's something that we could quickly add. Photometry data. If you send, I'll say for questions about what data formats Bento can take, just send me an example file and we can set up Bento support for those files. I'll say the Python GUI is a work in progress. It doesn't have the same plugin support right now. And we're working on making the experiment manager a little bit better organized to use it as sort of a database of all of your experimental data, tracking your animals over time. We'll run a tutorial on that a little bit later once it's a bit more established. But the process is similar, but the interface looks a little bit different when you're loading your data. Once everything's loaded, it looks pretty similar. If you have different start times for behavior videos and calcium imaging data, that's something that I think was in one of these files. Oh, do this quickly here. So if you have annotations and imaging data starting at different times, if you look at these check marks down here, there's a check mark for time offset between calcium and annotation start. So when you hit that, it adds a column called offset here. And if my imaging data starts before my annotation data, you want to add a positive number in seconds. If it starts after, right, other way around. If my behavior movie starts first, the offset is positive. If my imaging data starts first, the offset is negative, and it's in seconds. So you just add whatever that offset time is here and hit start import, and it will shift the calcium traces appropriately. The other thing, okay, so one thing I'm going to note with frame rates is that by default, Bento is going to trust the calcium imaging frame rate that we provide here. It could be that you have some dropped frames of your calcium movie and this isn't fully correct. So far, if you don't have timestamps for the individual frames of your calcium data, there's not much that we can do about it within Bento. If you do have timestamps, we can include that when we load the data in our population of the calcium time field. So if we go back and look at this experiment file for our mouse, this calcium time here is a timestamp of each frame in seconds. By default, it's just assuming whatever frame rate you provide is correct. But if you use something like the UCLA Miniscope that saves a timestamp for each frame of your movie, you can, if your timestamps are in the standard Miniscope format and they're in the same directory as your Miniscope output, they will be loaded and used as calcium timestamps within Bento. Again, this timestamp thing is one of the trickier parts of dealing with your data. And if you don't have a recording method that's really getting proper timestamps for when things are happening, it's tough to try to rescue it afterwards. I know some folks use tools like Bonsai that are really good for synchronizing things up, but I don't have a lot of expertise on the experimental side for how to collect data and how to get timestamps for your frames of data. I think that's it for questions in chat. Are there any other questions about the interface or analyses or data formats that you'd like to see in future versions of either the MATLAB Bento or the Python version? You can let me know here or send me an email. My email I'll enter in chat is annkennedy@northwestern.edu. Yeah, so message me there or via the GitHub issues page for Bento. And we will try to run these tutorials as we make changes and improvements to Bento and as the Python version becomes more developed. Also, if you have some coding experience yourself and would like to contribute support for a particular data format, send me a message and we can talk about how to get that integrated into the GitHub repo. So with that, thanks everyone who stuck around, and hope that it works out for you. Send me a message if you have any issues, and we'll take it from there. Thank you all.